Alright, so what we're going to talk about now is we're going to switch gears a little bit and move away from proteins and enzymes and talk briefly about lipids as a setup for the biological membranes that they come together to create. So by the end of this lecture series, this will be a two-chunk lecture series, 8A and 8B, we'll have a good sense of how and why cells are membrane bound and what the cells and the membranes are doing. So, the outline for both chunks is that we will begin by introducing and defining lipids, talk about what lipids are chemically, biochemically, and the different classes of lipids, and that's what the first chunk will be dedicated to. And once we understand lipids, we'll begin to talk about the structures they form, namely biological membranes and the membrane proteins that then exist in those membranes, and that will make up the second chunk, along with this idea of how those lipids and those proteins come together to create a membrane that adheres to a way of thinking called the fluid mosaic model. We'll end with some material in the next chunk that you're already familiar with, I'm sure, pumps and channels, passive and active diffusion. So here we see kind of a prototypical example of a eukaryotic cell. We have the cell itself, the nucleus, which contains the DNA, the Golgi body, the endoplasmic reticulum, the mitochondria, even a peroxisome is present on that diagram. And the one thing you might notice now in the context of this lecture that perhaps you didn't appreciate earlier is that every single one of these structures has in common the fact that it is membrane bound. So membranes are the defining characteristics not only of cells in general, not only do membranes make cells possible, but cells make organelles as well. The world out there is a dangerous place. We've got entropy driving most fundamental processes, so the universe is tending towards chaos. We've got all of this diffusion properties, this randomness, this heat, this energy, and so for a cell that can be quite a toxic environment to exist in. Additionally, all the chemistry that's needed to make life possible, all the chemistry that needs to occur within a cell, can be disrupted by all of this chaos and entropy that exists outside. Cells can encounter differences in pH. There can be protein and enzyme inhibitors. Perhaps there are the wrong concentrations of substrate molecules in the environment. And so if the cell is existing in this chaotic, entropy-driven universe, and we've got all these differences in variables and environmental conditions that we can't control, it makes the ordered, controlled, regulated environment within a cell very anti-universe, if you think about it. If the universe is a chaotic, highly variable place, and the cell is a very ordered, non-variable place, then the cell is kind of anti-universe. You can think of the cell as a bag of order, seeking its own existence within a universe that's driven by disorder. And so somehow we need a boundary. We need an inside and an outside. We need an ordered cell inside and a disordered universe outside. And that boundary, that protection, that delineation between chaos and order, that's the cell membrane. That's what the cell membrane is truly for. So today, again, the point of these two lectures is to discuss biological membranes, but before we do that, we have to, of course, understand their constituent parts, the lipids. So what is a lipid? Well, the fundamental definition of a lipid is based solely on solubility in water. That's kind of our defining commonality among all lipids. Lipids are, at best, slightly soluble in water, and most of the time lipids are completely insoluble in water. Lipids are insoluble because they're very hydrophobic, they're very nonpolar, they don't speak that watery language of charge, and so these things do not dissolve, they are not soluble in water. However, it's fair to say that because they're nonpolar and hydrophobic, we can get lipids to dissolve in nonpolar solvents, such as acetone or chloroform. So let's not say that lipids are never soluble, they're just very rarely soluble in water. The most common and typical lipids that we encounter on a regular basis are fats and oils. Those aren't the only ones, but those are the most common. As you know from personal experience, fats and oils are insoluble in water. They can be solubilized or dissolved in other nonpolar solvents, though. But even though we're using this definition of solubility for lipids, that really doesn't tell us much about lipids themselves, right? We can't define lipids as a biochemical molecule as an action it can't participate. how unsatisfying is it to say, well, lipids are things that can't dissolve in water? Nah, that doesn't really cut it. So we need to talk a little bit more about lipids to understand something about their chemistry. What can they do? What do they do? How do they behave in biochemical reactions? And when we begin thinking about lipids in that way, we see that they're pretty much a mixed bag. We can't and don't have a single unifying definition where lipids all do the same thing or all behave the same way. And so that's why we do default to the solubility definition, because in many ways lipids are very different. In fact, other than being hydrophobic, nonpolar, and not dissolvable in water, lipids can vary quite a bit from each other. So let's begin to parse out the differences of different lipids. We can classify lipids according to their chemical properties, and when we do, we can kind of come down to two broad categories of lipids. One of those categories are amphipathic molecules. Remember, we discussed amphipathic molecules before. These are molecules that contain both hydrophobic and hydrophilic parts. And so this class of lipids are usually made up of open chains of hydrocarbons. That's the nonpolar side. And then at the cap of that chain of hydrocarbons, carbon and hydrogen, we find more polar hydrophilic head groups. So here's an example of that type of lipid. This happens to be the fatty acid palmaic acid. I think I'm saying that right. And you can see that right here at its head group, we have a polar region, right? This is a hydroxyl group, so we have an electronegative oxygen linked to hydrogen. This is going to be partially negatively charged. The hydrogen is going to be partially positively charged. So this region of this molecule can interact favorably with water, and it does. It's hydrophilic. The rest of this, carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, down the line we go, the rest of this molecule is highly hydrophobic, can't interact with water at all, and so this is an amphipathic molecule. Drop it in water, and it'll form a micelle. Again, these are fatty acids. Triglycerols also fall into this large chemical group. The other class of lipids that we can define based on chemical properties are not linear, but they're ring-shaped. In fact, it's a collection of fused rings. And some of the compounds that fall into this category are steroids. Cholesterol is the precursor for most steroid hormones, so cholesterol falls into that category as well. And you see here what we mean by fused rings. We have 4 aromatic rings fused to one another, 1, 2, 3, 4, and we see tons of hydrocarbons associated with that as well. Lots of methyl groups, all of this very, very nonpolar. But look at this little bitty hydroxyl, one little hydroxyl sitting there. That's our only polar part of this entire molecule. The rest of it is completely nonpolar. In fact, we wouldn't call cholesterol even amphipathic because this is so minimally polar within the context of a much larger nonpolar molecule. So as we continue this discussion, we'll go in a little bit more detail of individual types of lipids so that we have some sense of how they differ and how they're important physiologically. The book spends 7 pages on this material. We'll distill it down to about 10 slides. So we'll start off with this fatty acid group. These are our classic lipids. These are the primary components of cellular membranes. As we've already said, they are amphipathic molecules. They have these polar, either charged or partially charged head groups that are capable of interacting with water and are hydrophilic, and then these very long hydrocarbon nonpolar tails that are hydrophobic and try to shield themselves from water. The polar head group on some fatty acids is a carboxyl group, and we saw that in the case of palmitic acid. So most fatty acids that are found in living cells, in living things, have an even number of hydrocarbons, and those hydrocarbons exist in an unbranched chain. We'll talk about branched chains in just a second, but here's an example of an even-numbered, unbranched chain fatty acid, such as palmitic acid. Here's that carboxy group, very polar, hydrophilic, interacting with water, and we see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, plus the initial carbon that starts the chain is 18 carbons. That's our even number. Some fatty acids are saturated, and what they're saturated with is hydrogens. In a saturated fatty acid, every single carbon is holding on to as many hydrogens through single bonds, as many hydrogens as it possibly can. You can think of it as a sponge completely soaked with water. Here it's a carbon chain completely soaked with hydrogens. In an unsaturated fatty acid, these hydrocarbon chains do not have the maximum number of hydrogens possible. The reason why these can exist without being fully saturated with hydrogens is because some of the carbons in an unsaturated fatty acid are engaged in double bonds. These double bonds are not naturally found in a cis configuration, meaning the fatty acid chain itself is on the same side of the double bond, and because this double bond is in cis, that double bond actually kinks the chain and makes it look this way. So now we can compare these two in their schematics. The fatty acid on the left is a saturated fatty acid. The fatty acid on the right is an unsaturated fatty acid. The fatty acid on the left has hydrogens at every conceivable spot, and no double bonds exist between the carbons. In the unsaturated fatty acid, we have a double bond between these two carbons. That means each of these carbons is holding onto one fewer hydrogen than it can, and that double bond, because it's in cis, kinks that fatty acid and makes it kind of take on this kinked or bent appearance. We can get a little bit of a better look at that by kind of seeing more of a schematic representation of these. Here on the top of this diagram represents a fatty acid like the one on the left, and you can see what we mean by saturated. Every single carbon is holding on to as many hydrogens as it possibly can. That unsaturated fatty acid is shown here below, just like the unsaturated fatty acid on the right. And you see, because of this double bond here, these two carbons are holding on to one fewer proton each. There's a proton missing from here and a proton missing from here in order to make that double bond possible. Because we are in cis, and we have each of those hydrogens on the top of these carbons, and below those carbons we see we have the open space, these two protons sterically interfere with one another and cause a kink in the fatty acid chain. So I want you to ask yourselves, from your own empirical knowledge, which one of these are healthier for you, saturated fatty acids or unsaturated fatty acids? In your normal day-to-day activities, which one of these do you know to be healthier? Hopefully you're aware that unsaturated fatty acids are the better fatty acids. If you have to consume fatty acids at all, or fats, you prefer unsaturated fats to saturated fats. You're about to see biochemically why that is. As you can probably see already, unsaturated fatty acids can't pack as densely together as possible. That kink makes them take up more room. Let me ask you to imagine something, if you would. Imagine that you have 5 people standing shoulder to shoulder, one right next to the other. So they're all looking forward, and they're standing as close to each other as they can, one after the other, shoulder to shoulder. Now imagine that each one of those individuals sticks a leg out. What's going to happen? You can imagine that now they have to spread out. They can't pack shoulder to shoulder anymore, because as they each spread out one leg, as they each kick one leg out to the side, let's say it's their right leg each, they need more room. They fill more room because of that leg sticking out. So now each one of those people have to stand further apart from the others, and they can't pack as densely together. The same thing is happening in unsaturated fatty acids. Because the fatty acid is unsaturated and has that double bond-induced kink in it, that fatty acid takes up more physical space than it would have otherwise, and so it stops those fatty acids from sitting or clumping as tightly and densely together as they normally would. This makes unsaturated fatty acids have lower melting temperatures. That means they become liquid at higher temperatures. And saturated fatty acids work in the opposite way. Saturated fatty acids are those 5 individuals standing next to each other with arms strictly at their sides and legs straight down. You can pack those 5 individuals up together quite densely. No kink, no legs sticking out, no problem. You can pack those fatty acids in deep. And with that higher density comes a higher melting temperature. These fatty acids tend to stay solid at room temperature, not liquefy. So let's take a look at this. This is olive oil, near and dear to my heart. And what type of fatty acid do you think olive oil is primarily made of, keeping everything in mind that we've talked about? Hopefully you said that olive oil is unsaturated, and much of olive oil is unsaturated. All of those unsaturated fats, with all their legs sticking out, all their kinked fatty acids, are taking up more space and packing less densely. Less dense things are liquids, not solids, And so olive oil is made up of quite a bit of unsaturated fatty acid. Compare that to this. Also a fat, but what kind of fat do you think butter is primarily made out of? Hopefully you said saturated fatty acids and you'd be right. In butter, those fatty acids are more linear and can pack more densely together because the kink is not there. And since they can pack more densely together, they stay solid at room temperature. Now we know empirically that olive oil is healthier to cook with than butter. Butter's so good, but it's not so healthy. But now we understand why. The chemical nature of the olive oil fats means that it's less dense. Well, less dense in olive oil also means less dense in your arteries. And so as these fats do somewhat collect in your arteries, they don't stick tightly together and they can't really grab a foothold. They keep flowing with the blood because they are less dense. More dense fatty acids, able to be more dense because they have no double bonds and no kink in their tail, can not only stay solid at room temperature, they can also pack more densely in your arteries, potentially creating blockages there that can cause bigger trouble and heart disease later on in life. If you don't believe me, just look. In the average teaspoon of olive oil, we have 14 grams of fat. Of those 14 grams of fat, only 2 grams are saturated, and the other 12 are various forms of unsaturated fatty acid. In a tablespoon of butter, we have 6 grams of fat. But of that 6 grams, over half are saturated fatty acids, far more percentage-wise than the fatty acids we see in olive oil. So just as butter stays solid at room temperature because of the density allowed from an unkinked tail, saturated fatty acids tend to remain solid, if you want to think of it that way, in your arteries, leading to blockages. Unsaturated fatty acids are far more fluid and less dense because of that double bond kink in their tail that allows these fats to flow through your arteries more easily, clumping up and glumping up together less in those arteries, causing less blockages, and therefore unsaturated fatty acids are far healthier. With all that in mind then, what in the world are trans fatty acids? What we probably know about trans fatty acids so far is that they are the least healthiest of the bunch. In fact, New York City, under the leadership of Mayor Bloomberg, banned trans fatty acids from all fast food restaurants across the city. But what are they and what makes them so bad? Well, if you think about it, unsaturated fatty acids are healthier. We just proved that biochemically. But man, oh man, is butter so good. Saturated fatty acids just taste better to us. If that's the case, food scientists, corporations, decided a long time ago, can we get something that looks healthy on a label, but is unhealthy and tastes good so people will like it? In other words, is there any way we could take the labeling benefits of unsaturated fatty acids, because everybody thinks those are good, but make them behave and taste like saturated fatty acids, which is what everybody likes? And they did it. In fact, what these scientists did was they got some normal unsaturated fatty acids, like olive oil, and they artificially saturated them by forcing hydrogens to go wherever they could. Then they removed other hydrogens and achieved the synthesis, the artificial synthesis of these trans fatty acids. One look at a trans fatty acid, and you can not only understand why it has the name that it has, but you can also understand how it can look healthy, but be unhealthy. Here is our old friend, a normal unsaturated fatty acid, remember, in the cis configuration. Here is the double bond. The double bond induces a kink, because the protons are in cis and they're pushing away from each other. When those scientists forced protons onto this molecule and then allowed some other protons to fall off, they created this. This is an unsaturated fatty acid with a double bond right here. We know it's unsaturated because there's a proton missing from here and there's a proton missing from here. On a food label, especially in the 80s and 90s, this would be listed as an unsaturated fatty acid. And why shouldn't it be? It is, by definition, unsaturated. But it's a trans unsaturated fatty acid. Trans because this proton is going up and this proton is going down. And because these protons are in trans, hence trans fatty acid, this is not a kinked fatty acid, it is linear. So what was it about the saturated fatty acids that allowed them to pack densely? Do you remember? It was the linear nature of their fatty acid tail. Because the fatty acids of saturated fatty acids were linear, they could pack closely together. Well, these fatty acids are linear too, and they can pack closely together. From a physiological standpoint, there is no functional difference between saturated fatty acids and these trans fatty acids. They both can pack very densely, they both can clog arteries, they both stay solid at room temperature. But on a label, this used to be listed as unsaturated because by chemical definition, that's what it is. It contains a double bond. So food corporations would add these trans fatty acids to their foods so that they could list a lower amount of saturated fatty acids, a higher amount of unsaturated fatty acids, and the consumer would look at the label and says, oh, this can't be unhealthy. It's got a lot of unsaturated fatty acids in it. But in fact, these unsaturated fatty acids were behaving like butter. They were behaving like saturated fatty acids and creating the same health problems. So if we look at it a little more schematically, this is a typical unsaturated fatty acid, nice and straight and linear because every single carbon is fully protonated. This is a typical unsaturated fatty acid with this very pronounced kink in its tail, causing it to be less dense and healthier. It is kinked because these protons are in cis. And here is a trans fatty acid, unsaturated because of the double bond, but the protons are in trans, giving the overall tail a very linear configuration, making it just as unhealthy as butter. Why human beings will do these things to other human beings, I will never understand. How the profit margin of a food corporation could trump the health of all the consumers eating those foods is beyond me. And wait, I misspoke. Because when I just said that all of this made trans fatty acids just as unhealthy as saturated fatty acids, I was wrong. Because the fact of the matter is that trans fatty acids do not exist in nature. These were man-made and woman-made. And so trans fatty acids are fully artificial. That means we do not have any enzymes to break down these trans fatty acids. So these trans fatty acids are unmetabolizable in our body, making them accumulate even more than butter and have that much of a greater risk of causing plaques because they can't go anywhere. So we've created these trans fatty acids that are not only as unhealthy as butter, but more so because our body doesn't even know what to do with them and so largely ignores them and leaves them in the blood. That's why there's been such an uproar against trans fatty acids. And that's why trans fatty acids have been banned in some venues and in some jurisdictions. So fatty acids themselves, even the healthy ones, even the normal ones like cis and saturated, are rarely found free-existing and free-floating in nature. They are part of larger structures. And so the question becomes, what are they part of? And they are, of course, part of larger lipid-based structures. Larger and larger subunits that come together to eventually form things like cellular membranes. But what are some of the other lipids that these fatty acids can interact with in order to create the biomolecules we need for life? Well, one of the most important other lipids that we need to discuss in that context are triacylglycerols. Triacylglycerols are relatively small molecules made up of a single glycerol molecule and having the potential to interact with 3 different constituent parts. Now, those could be 3 different fatty acids, and if we did indeed make that, we would have a triglyceride. This is a triglyceride. We have the glycerol here, making 3 bonds, and the 3 bonds we're making is with 3 independent individual fatty acids. In this case, each of these fatty acids is saturated, hence it being very linear. That doesn't necessarily have to be the case. And we would refer to this as a triglyceride. Tri because there's 3 of them, glycer because this is a glycerol backbone, and ide meaning all of the same. Again, this is built on the glycerol backbone, But we don't have to create a molecule like this. We can create a molecule where all of the fatty acids are different. We can create a triglyceride where 2 of the fatty acids are the same and 1 is different. Regardless of how we mix and match these, triglycerols are amphipathic because we have a very hydrophobic backbone. We have a hydrophobic bridge here of the glycerol itself, but we also have these polar regions, the polar head groups of the individual fatty acids. Triglycerols are fat storages, the adipose that contributes to our having fat reserves for energy purposes are largely triglycerides, but triglycerols are not found in the membrane. When it comes time to release these energy stores from these triglycerides, we can only harvest energy from individual fatty acids, and so we need special enzymes called lipases, and lipases can cleave and release individual fatty acids from these triglycerides, and then those individual fatty acids are metabolized through a process called beta oxidation, but we won't cover beta oxidation in this course. And incidentally, soap, you may have heard from history classes or stories that you've read in school that soap used to be made from whale blubber, soap used to be made from fat, and indeed that's true. Soap is made from the released glycerol that occurs after we have the fatty acid release. So soaps are usually made up of these glycerol backbones. People would harvest whale blubber, treat it with lipase, and skim off the top and use that for the soap. That was the glycerol backbone of these triglycerides. Sometimes we can use glycerol and not make a triglyceride. Instead, we can use those 3 slots in other ways. Oftentimes, 2 of those slots will be taken up by fatty acids, just as we saw before, but the 3rd slot can be taken up by phosphoric acid instead of a 3rd fatty acid. When we do that, we create a more polar molecule, a molecule that has a much more polar head group, and we call that a phosphatidic acid. So here's an example of a phosphatidic acid, and one look, you can see why this is much more polar, much more amphipathic than the triglyceride we talked about a moment ago. We do have 2 hydrophobic tails, large linear tails of hydrocarbon, but the phosphatidic acid component here, this phosphorus group, is charged. So it's very, very polar and very hydrophilic. Here's another schematic representation where these R1 groups are meant to symbolize just a very, very long hydrocarbon tail that's coming off of this. So 2 fatty acids coming off the first 2 slots with the 3rd slot filled with this phosphorus group. Sometimes we can take that phosphoric acid group and link it to yet something else. Oftentimes we link it to an alcohol. And if we do that, we make a phosphoacyl glyceride. Phosphoacyl glyceride is telling us that we have a phosphate group and an alcohol group all linked together with a glycerol. And if you know enough about glycerol, you know that the other 2 slots are held by a fatty acid. So this is a phosphoacyl glycerol. We can see the first fatty acid in the first slot, the second fatty acid in the second slot. They're holding on to glycerol, which is serving as the backbone. That 3rd slot of glycerol is making interaction or a bond with the phosphatidic acid. But now that doesn't end in a hydroxyl group, that ends with a linkage to an alcohol group. And so this is our phosphoacyl glyceride. Phosphoacyl glyceride. And this bad boy is not only highly amphipathic because of this very charged and polar head group, this is the primary component of a lipid bilayer. This is what our cell membranes are made out of. Now there's a lot of variability in this group. We can change the alcohol, we can swap out different fatty acids, so we can have dozens, if not hundreds, of different types of phosphoacyl glycerols, but all of them share the similar structure of 2 fatty acids on a glycerol with a 3rd slot making a contact to a phosphate group that is then linked to an alcohol. Again, the primary component of the lipid bilayer. Just to complete the story of lipids before we wrap up this chunk, we also have waxes that belong to the lipid group. Waxes are a completely different group of structures, very, very complex, made up of very, very different types of molecules. But of course, if they're lipids, they all share the characteristic of being insoluble in water due to them being nonpolar. Some examples of waxes are here. You can see they're very complicated chemical structures, quite large. We're running methyl groups of 14, 15, 24, 29. And for the most part, waxes occur naturally as protective coatings. We see them largely on fruits, and some animals will coat their fur with waxes or their feathers with waxes to largely make them waterproof and keep the animal from getting physically wet so that it gets cold. So stems, fruit, leaves on a plant are protected, fur, feathers, skin can be protected. We obviously make wax in certain parts of our body, and that wax is meant to trap foreign materials from entering too far into our ear canal. Another lipid group unto itself are the sphingolipids. Those of you who are neuroscience majors may have heard of these before. This is the very unique and specialized fat component which insulates neuronal axons. It makes up the myelin that insulates the axons of our nerve cells. There's no glycerol in a sphingolipid. It's similar in structure to other phospholipids, with the exception of missing glycerol. It has a polar head group and a nonpolar tail, so it's very amphipathic. And then we have glycolipids, these weirdo hybrid structures where a sugar is actually bound to the alcohol group of the lipid. So it goes fatty acid, phosphate group, sugar, alcohol and sugar. And most of the time that sugar is either glucose or galactose. And these play largely signaling roles. So sometimes cells will talk to one another using these sugar-fat hybrid molecules. They're also found in the membrane of some of the cells in the brain, marking them as different and protected so that some molecules can't diffuse into brain cells. So that's pretty much it for this lecture. We talked about the purpose of a cell membrane in the very beginning, that this is a protective coating of a cell to keep it kind of shielded and boundaried from the entropy and the harsh climate that occurs outside of a cell. And we said that these cell membranes are made up of lipids, so we better get our lipid act together before we move ahead on cell membranes. And we did that by talking about lipids very much in general, defining them by their solubility in water. And then we went on to talk about different chemical classes of lipids, fatty acids, and the fused rings. We spent most of our time today on the fatty acids, of course, because there's so much meat there, as we talk about saturated fatty acids that are saturated with hydrogens, meaning every carbon has as many hydrogens on it as possible. Moving on to unsaturated fatty acids that contain double bonds. Those double bonds mean that some hydrogens are missing, and normally in a natural unsaturated fatty acid, that double bond is in cis, introducing a pretty pronounced kink into the tail of that unsaturated fatty acid. We use that as a jumping off point to kind of digress a little bit onto trans fatty acids, just as an interesting side note of where these things can go, and how a strong biochemical background can really make you an expert in your own life and your own personal decisions. And once we understood fatty acids, we could use them to build larger molecules, such as our triacylglycerides for fat storage and energy storage, but much more importantly, our phosphoacylglycerols. These are molecules that have a glycerol backbone, contain two fatty acids of any number of types, and that third glycerol slot is consisted of a phosphorus group or a phosphatidic acid linked to an alcohol. Very, very polar head group, very, very nonpolar tail group, and so these are the phosphoacylglycerols, the primary fatty component of our cell membranes. These are the molecules that make up our lipid bilayers. We'll talk a little bit more about it in the next lecture, but essentially those nonpolar tails are huddling away from water on the interior side of the membrane, and those polar hydrophilic head groups are existing happily on either end of the cell membrane, interacting with the watery environment that makes up the cell, as well as the exterior environment of the cell. So since the inside and the outside of the cell are very watery, that's where the polar head groups are happy interfacing with, and they can shield their fatty acid tails away from water, keeping those fatty acid tails on the inside of the membrane. Again, we talked about saturated fatty acids being less healthy because of their straight and unkinked tails. They can stack more densely and stay solid at room temperature, stay solid in our arteries. And the unsaturated fatty acids tend to be less dense and kinked. That allows them to be more fluid, not only in our arteries, but also in the membrane itself. So we'll leave it at that for this lecture. We'll pick up with the second half of this lecture in the next chunk, where we'll spend quite a bit of time talking about lipid bilayers in general, now that we know what they're made up of. We'll talk about membrane proteins, that fluid mosaic model to describe how all of these components interact with one another, and we'll close that out with some discussion of cellular transport across the lipid bilayer. Until then, thanks for watching.